Thanks everyone for coming today. And thanks also to Kelsey for sharing firstly the podcast, which was wonderful, and agreeing to do this a week later. The research Kelsey's involved with is really, really fascinating, especially to us sheep addicts that are all here. And yeah, we just can't wait to hear sort of a bit more of an in-depth presentation, see some graphs and some charts and stuff. And yeah, looking forward to hearing it.
Yeah, perfect. Thank you, Sophie. Like I said, this is a very kind of condensed down, brief presentation because I didn't want to spend all of my time just kind of rehashing some things that we talked about in the podcast, but it's just going to kind of give us a more visual of maybe some things that were discussed. So with that, let's get right into it. So my research background, as I previously discussed, is using estimated breeding values through the National Sheep Improvement Program. And mainly that is going to be the post-weaning fecal leg count estimated breeding value. So the reason we use the post-weaning value is that that value is actually normally a little bit more accurate than the weaning value, being that around weaning, we haven't really seen a large parasitic burden within those individuals yet. So that's just the reason why we choose to use the post-weaning fecal leg count, EBV. And from this, it gets to be a little kind of in the weeds, but we can see that negative values are favorable for a post-weaning fecal leg count EBV. So in an ideal world, we have two rams here where we have ram A who's sitting with a post-weaning fecal leg count EBV of a negative 99. We would consider him to be a parasite resistant individual, meaning that he's going to pass along genes of resistance to his potential offspring. And throughout the presentations, you'll hear me refer to him as a low catodon, being that he's incredibly negative in terms of that value. On the opposite end of the spectrum, we can see Ram B is incredibly positive in terms of his value. Therefore, instead of having percent reduction, he would actually have percent increase in terms of worm egg counts. Therefore, we would consider him to be passing along genes of susceptibility, and I'll refer to them throughout this presentation as a high catodon. So for the research framework, throughout this presentation, color is going to be incredibly important. So you can see that I have in green individuals that are going to have a post-weaning fecal leg count EBV of less than negative 50. So for my work, we would consider those individuals to be parasite resistant or low catodons. On the opposite end of the spectrum, anything that has a post-weaning fecal leg count EBV of greater than positive 50, we would consider to be parasite susceptible or high catodons. And then a little bit later into the presentation, we'll actually get into that middle ground of what exactly do we call the sheep that aren't on the extremes of that spectrum. What about the individuals that have a post-weaning fecal leg count of between a negative 49 and a positive 49? And so I'll call them kind of intermediates, or they'll be right in the middle, so the mid-catodon group. So getting right into the actual data now, I talked a lot about the depth loss and kind of how this was the pivotal starting point of my master's work. And you can see I arrived in 2019. So we were just coming off the cusp of this really large death loss here in 2018. But when we separated that death loss out between whether you were sired by a low post-weaning fecal leg count EBV sire, so you were a parasite resistant sired lamb, or you were a parasite susceptible sired lamb, we see lots of variation within that death loss percentage. Now, we can see that this is a pretty intensively managed operation at the Southwest Agricultural Research and Extension Center down in Glade Springs, Virginia. So that 10% death loss is actually pretty on par for this particular production system. However, so consistently low PFEC sired sheep reach right around that 10% death loss, whereas there's a lot of variation depending on pathogen load within the particular environment, especially when we're talking about the high lambs or the lambs that come out of susceptible sires. So that's then going to bring us to the breeding scheme or the milk kind of component of this. And I talked about that originally we really didn't know what to expect. So we only chose to report on kind of 20 individuals of each group. And you can see I have some criteria over here on the side because we know that age and the amount of lambs in which she produces is going to impact IgG from a colostrum sense. So we wanted to ensure that our ewes were relatively similar outside of the one thing which we were divergently selecting them for, and that would be post-weaning fecal egg count. So you can see we put age, a Katahdin hair index, which is just going to kind of be a maternal index through NSIP, and also lambing requirements on these ewes to ensure that we were getting ewes that were consistent outside of being divergent in terms of their post-weaning fecal egg count. And then we collected 3 samples on those ewes. So one would be early, so that would be a colostrum sample. The other one would be a mid sample, which was approximately 30 days post-partition. And then finally a late sample, which was approximately 60 days post-partition. And so those would both be when we're talking about milk. So that would no longer be colostrum. Now, getting into the data that we saw from this, you can see that we were really, really excited because at every single time point in which we sampled, low PFEC or parasite resistant Katahdin ewes consistently had more IgG within their colostrum. But probably more importantly, at that early time point when mom is passing passive immunity to her offspring through colostrum in that early sample, there's a 2.5-fold difference in terms of IgG. So we could not even begin to expect that type of value. And that kind of got us thinking, okay, well, we have to do this again on a larger scale. But there was actually something that I didn't get to talk about in the podcast, simply because it was a little bit harder for me to measure, and that was actually color. So I had to sit down and dialyze these colostrum samples and milk samples that we were getting. And there was one thing that I noticed as I was sitting down and dialyzing this colostrum, and was that one group in particular had consistently more yellowness and density within their colostrum when compared to another. So on the right here, you can see I have whole cow's milk just to give us a good reference of something that you could pull out of your fridge and look at. And then I have the kind of a composition of all of the colostrum samples of those ewes combined into 150 ml tube. And we can see that the low ewes or the parasite resistant ewes have more yellowness there to their colostrum. So that really got us also thinking like, that's an interesting dynamic. So what I had to do as a researcher is find a way to measure that. So for year 2 of the project, we said, all right, we're no longer going to care about the 30-day or the 60-day milk sample. We're only going to focus on colostrum composition. And then I also did some colorimetry. It's mainly used in food science applications to give like redness of meats or yellowness of cheeses. But all you need to know is that's essentially what I did to give me a numeric value so that I can run statistics and definitively say what my eye and your eye is seeing, that there was in fact color differences between the colostrum samples of these groups. So now we're going to look at colostrum in all of the ewes out at the Southwest Agricultural Research and Extension Center. And so that's not only going to encompass low Katahdin ewes and high Katahdin ewes, but now we're also going to grab that group in the middle that's maybe kind of intermediate in terms of their resistance to see what their colostrum looks like from both an IgG standpoint, but also a color standpoint. So getting right into the color, we can see that parasite-resistant ewes consistently on average had more yellowness to their colostrum when compared to both that intermediate group and the susceptible group for parasite resistance. Moving right along, when we're talking about IgG, though, we get this really nice stair-step effect in which resistant Katahdin ewes, or those that were low in terms of their post-weaning fecal egg count EBV, on average had significantly more IgG when compared to that mid grouping. And then finally, if you were in that high grouping or you were a susceptible ewe in terms of your genotype, you had a relatively low amount of IgG within your colostrum when compared to all 3 of those groupings. So really, really cool kind of stair-step effect that we see within those 3 groupings.
Just quickly, Mark Dawson, did you have a question? It's saying that your hand is up. It might be a mistake. No, okay. Carry on.
So, I talked about that early sample, right? So this is just a bar graph of the exact same line graph that I showed earlier, but now instead I kind of pulled out only the colostrum sample. So in year one, average IgG for the 40 U's that we analyzed looked a little something like this. Well, wouldn't you know, when we go back and we analyze IgG, across the entire flock and I look at those two similar groupings in terms of whether you are low in terms of your post-weaning fecal leg count EBV or you are high in terms of that post-weaning fecal leg count EBV, we see a graph that looks incredibly similar. So this was just so biologically relevant that we could have consistency of measurements depending on whether we were only sampling 40 U's or if we were sampling all of the U's at this particular production facility. We see very, very similar results across year, which was very, very telling that I do believe there's something of biological relevance that is occurring within these U's. So that was all of the colostrum project. So now switching gears a little bit to talk about, all right, so mom is funneling more antibody within her colostrum to lamb, but now we wanted to see if lamb was doing anything with that antibody. How is that lamb going on into their productive life? And in order to do that, we needed to give them something to mount an immune response to. And so we thought to ourselves, what a good way to kind of measure a lamb's immune response to something, with that being a vaccination that was already occurring within this production system. So this is kind of just going to give you a timeline of the traditional CD&T vaccine that we would give to these sheep out at the Southwest Agricultural Research and Extension Center. And you can see that in combination with the CD&T, so a management strategy of having a type A vaccine, because that's what the lambs were passing away from in that 2018 death loss. So that was actually implemented as a management strategy, and we saw death loss return to normal after that mitigation strategy. So that was also administered. So we had a CD&T, which included Clostridium perfringens type C and D, along with a type A vaccine. So lambs would be born mid-March to early April. Those lambs would get to be kind of on average as a cohort around 8 to 12 weeks of age, or excuse me, 6 to 8 weeks of age. And then we can see that they would be weaned, followed by a booster 4 weeks later. So, for those of you that don't know what a traditional response to vaccination is supposed to look like, I'll go ahead and spoil it for you that the parasite resistant lambs or the low post weaning fecal leg count EBV lambs do a beautiful job of it. So, we can see upon initial vaccination, those lambs' cells will go out and they'll sample parts of that antigen and they generate a little bit of serum IgG to that initial vaccination and then return to a relative baseline right around that 4 week mark. But then upon booster vaccination at that 4 week point, they are able to rapidly intake that antigen and generate huge amounts of amnestic antibody, indicating that they have a protective immune response. Now, let's talk about the problem. So we can see that the high PFEC lambs, or those lambs that would be parasite susceptible, never really generate a huge response to that initial vaccine. They kind of remain relatively low. There's a slight peak maybe at right around that kind of 2 week point, but because they never quite generate antibody to initial vaccine, they cannot nearly get to the same level of protective immune response at week 5 from the booster vaccine. So we can see huge disparities there between those groups immediately following vaccination. And this is kind of indicative of maybe parasite susceptible sheep would actually require a third booster in order to incur the same amount of protective antibody as parasite resistant individuals. And so this is probably one of my favorite things that I love to show producers, because I've talked about, passive immune response. I've talked about active immune response, but, all of that is fine and dandy. At the end of the day, sheep are still going to get sick. Just because we select for sheep to be healthier does not mean that they're never going to encounter a pathogen presence within their lifespan. So essentially what we did for this project was we gave them some external components of gram-negative bacteria called LPS. And you can see that those sheep will respond like they have really a respiratory infection. So think weeping of mucous membranes, fever, coughing. They'll be ears down, heads down. They'll even do a slight phlegming. They'll be very uncomfortable during all of this. So I like to show producers these videos because essentially what it does is it gives some merit to this, and it's not me just standing here showing you a graph. It's you being able to analyze this with your own eyes and tell me which pin you think appears sicker. So this is 3 hours after that LPS exposure, and I'm going to start the video now. And what you can see is immediately all of the parasite resistant or the low PFEC Katahdin ewes are at least standing. They're kind of, their ears are down and you can tell they're not feeling great. However, when Hannah goes to enter the pen, every single one of those sheep will at least respond to her stimuli. Now let's talk about the parasite susceptible group, or those ewes that are incredibly high in terms of their post-weaning fecal egg count. They feel so physiologically ill that when she bends down to touch them, they will not even respond to her stimuli. They're laid out lateral. Their grimace scores are incredibly high. So when we're talking from a treatment perspective, you tell me, if you were to walk out to your pen of ewes and you saw one that was kind of laid out lateral in the pen like this one, which one do you think is more likely to receive that antimicrobial treatment? The high ewes or the low ewes? And I think all in all, while they're both experiencing the same type of challenge, the low ewes for some reason are better able to adapt to that challenge and kind of move on from it. Whereas we see the high ewes really succumb to challenge, and therefore they have very severity of sick behaviors and illness. So what we can say from my research right now is that selection for low PFEC, EBV, and Katahdin sheep, so those sheep that would be parasite resistant, we also see a reduction in lamb death loss. We have ewes that produce more IgG within their colostrum, sheep that respond better to vaccination, and also sheep with less severity and duration of sickness whenever we do challenge them with those external components of gram-negative bacteria. So that's pretty much all of I have. I want to open up the floor to questions and kind of get some feedback from all of you here in the room. And let me actually go back to, so with all of that being said, thoughts, comments, concerns?
Any questions out there? Surely there is.
I suppose I'll start. How, obviously this is Katahdins, how does this apply to the other breeds and what we've got over here?
So, I actually forgot to mention what I was going to mention about those videos, but if you go back and look at that video, you'll see that the group of the low Katahdins that I was talking about, that would be the parasite resistant type of phenotype, those ewes appear to be more white and have less of that kind of wool along their back. And therefore, so the whole kind of hypothesis here is that when we're digging back into selection for post-weaning fecal egg count EBV, we're actually digging back more into genes related to the St. Croix, which would be an island breed that we know would have developed some resistance to that pathogen just from being around Homunculus almost year long. So there was some natural selection there put in place for the Croix. So the whole idea is that maybe when we're selecting for this, we're actually reaching back into genes of the Croix. Whereas if you go and actually analyze kind of the makeup of those high sheep, there's a lot of them that will have wool along their back. They're more likely to have kind of specks, and they're more likely to be more colored up. So therefore, we think we're actually reaching back into genes of susceptibility, much like either the Tunis or the Suffolk. So the Katahdin is in fact a composite. So there's a lot of hodgepodge things that are going on a genetic level. But we think that by selecting for post-weaning fecal leg count EBV, we may be kind of trending back towards genes that are kind of segregating based upon those breeds that went into the initial mating.
Is there any work going on in different breeds, Kelsey? I can't remember if we asked that in the podcast or not. But
not right now. So the unit that I was talking about in Virginia Tech, they have that beautiful divergent mating in which they actively breed for one group of sheep every year to be incredibly low in terms of that value or incredibly negative. And then they actively breed for one group of those sheep to be incredibly positive in terms of their post-weaning fecal leg count EBV. So that divergent mating really gives us a beautiful research setting. Now, do we see a lot of that at other places? No. And my whole goal is to start to get some of the polypay sheep, which would be more of a maternal wool breed here at Kansas, get those kind of to be segregated out like we did with the Katahdins down at Virginia Tech. And hopefully we could start to maybe do some research in breeds outside of the Katahdin. Because yeah, right now we do have to confine this to the context of Katahdin sheep. I'm not quite sure if we were to look at response to vaccination in merinos, what they would do. We don't really know because no one's out there actively doing it, but we're not actively doing it because you don't have that nice divergent mating set up. So hopefully, even if we don't have the divergent mating, we can at least start to maybe explore immune responses and look at kind of things that are going on from a physiological standpoint and use within an NSIP, because I feel like that at least gives us a good starting point for seeing how other breeds would do in response to CD&T.
Can I just ask a question? It's really interesting work you're doing, Kelsey. So what is the next thing you're going to try and find out about the effects with worm resistance and other things that it may also benefit?
So my whole, I think my career, I'm really intrigued by the colostrum. I think that colostrum actually, we have no idea why that colostrum is more yellow in color. I actually did a correlation because I wanted to be able to tell producers when you see more yellowness within colostrum, you have more IgG there. But unfortunately, it didn't pan out that way. The correlation didn't work. So therefore, something is undermining that yellowness. And I'm not quite sure what it is. It might be some vitamin A kind of synthesis that's occurring there. So some vitamin A protagonist that will then allow that sheep to go on and generate more vitamin A. I'm not quite sure. It could be something as simply as there's more fat in it. So I think my next steps, because the colostrum samples are still there from that particular unit. And so I don't necessarily have the type of sheep that I would like in Kansas right now to be able to do really cutting edge research. So therefore, I think the next step is let's do some metabolite analysis of colostrum. Let's see if there's certain metabolites that are at play that's maybe driving that color difference, or even Brix values. We played around with the Brix values a little bit and found that what the Brix values were in terms of Katahdin sheep were not necessarily consistent with some other literature that was out on traditional wool breeds. So maybe the Katahdin has a different kind of metric for that Brix value as of if we can have a shoot side measure of IgG within colostrum, that would give producers a really good way to select for very high quality colostrum, not only from a nutritional sense with the metabolites, but maybe even an immunity sense or an immunological sense. So I say a lot, but like I could ramble on about the colostrum for forever, but I think that's my kind of next steps. While I love the response to vaccination, I think the long-term goal is I could spend the next 30 years just analyzing colostrum and looking at how the programming of tissues, when we talk about epithelium, like what type of receptors or antagonists are having certain metabolites cutting on within those individuals, and then how does that affect their lifetime productivity?
Can you see the specific colostrum quality EBV in the future?
Maybe. I would like to see one. I'm not quite sure if we're there yet. I know that a lot of producers are really concerned with like the maternal weaning weight in milk, which is to be fair. But in an ideal world, I would like to have some sort of a measure of colostrum. But the colostrum is harder to measure. As I stated, you have to dialyze out all that colostrum. So it's quite a labor of love. But I say all of that to say that, yeah, I think that that is the starting point of that lamb's trajectory in terms of its entire lifespan. So if we can kind of maybe unravel some of the nuances to colostrum, we might actually be able to take a parrot, like genetically, let's say that we have a lamb that we know should be parasite susceptible. Well, what if I give that lamb parasite resistant colostrum? Does that then impact their ability to go on and combat parasitic infections? We don't know. So you could actually start to do some cross fostering where we start to supplement lambs that are genetically one phenotype with colostrum from a ewe that's a different phenotype and see what occurs.
Question there from Kelsey Keeling in the chat. Do you have recommendations for selecting parasite resistance if you aren't in NSIP?
Yeah, that's a troublesome one. You can see that all of my research centers around NSIP, and I think that from a genetic sense, it is by far the way to go. But that, in fact, does require a lot of time, and it's also a factor of cost. But for producers who maybe aren't enrolled in the National Sheep Improvement Program, I recommend fecals. I think that fecal egg counting is a really good way for you to set up some culling metrics. And even if we're not willing to go as far as fecals, even just doing a 5-point check or FOMACHA scoring, really good ways to record individuals that are not doing well for your particular flock and cull them. Make those culling decisions on what we see from a physiological standpoint. I get that sometimes I'll have a producer that calls me and they're like, well, that's my best ewe, you know, she's done X, Y, and Z for me. However, if she's consistently at a low FOMACHA score and consistently is a struggle in terms of her body condition, then we need to be recording that. So therefore, when it does come time to make those hard decisions, we actually have a record and we can go back and remind ourselves of all the trouble we had with her up until that point. And then that makes it a little bit easier for us to then make that decision and say, yeah, we probably need to cull this one.
Kelsey, we had a chat about resilience after we stopped recording. And so I thought it'd be good to chat about that now. What are your thoughts on like all this? Obviously, there's two ways people think about resistant or dealing with parasites in sheep. One is just like for resilience, one for resistance. What are your thoughts on that?
Yeah, so as an immunologist, I'm probably not a good one to talk to about it. My training is once again that we just need to select for resistance. Resilience is only going to exacerbate the problem, and therefore you don't really make much forward progress. So I'm probably not a good one. You'll go and talk to 5 different people, and they'll all have 5 different takes on it. My research is obviously very centered around genetics and very centered around either parasite resistant or susceptible individuals. So therefore, I'm going to kind of stay of the camp of I believe we should genetically be selecting for parasite resistant sheep, and therefore we won't have to deal with the issue of resilience.
Yeah, good. Just need to record saying that, so that's good. Excellent. The Linton's asked, is it likely we'll find markers that will enable genomic selection?
Oh, yeah. So we did a GWAS. My good friend Jake Thorne actually helped me a whole lot with that. So I'm not a geneticist. As you can see, I do a lot of immunology, and I kind of just dabble enough in the genetics. But we did do a GWAS study on a very limited amount of Katahdin sheep. So I want to be very careful saying this data, but we did. We wanted to see if there was any SNPs that were associated with kind of resistance or susceptibility within Katahdins. And what we found that there actually was a taste receptor, which is an interesting thing to think about, because your taste receptors not only are just on your tongue, but they line your gastrointestinal tract. So if you have this particular taste receptor, are you more likely to sense parasitic antigen? Like, can you taste the parasites better than other sheep? So that was another whole little kind of rabbit hole that we went down, that I didn't even put in my dissertation. Because once again, I'm not huge on the genetic side of thing, and running studies like that, looking for those SNPs, I take a lot of time and a lot of effort. But we did in fact find a particular taste receptor that we thought was interesting just from a kind of physiological standpoint. And yeah, we haven't really ran with it, but just some food for thought in the future. If someone does want to pick up, maybe I need to collaborate with Jake again at Texas A&M, and we need to get to looking at this particular taste receptor, and if maybe that's also segregating with either parasite resistance or susceptibility.
For those who just to NSIP, so that's similar to the same thing as sheep genetics or SIL in New Zealand and Australia. So that's the American version of the performance program, just to sort that acronym out. But Mark Ragg said, did you find any surprising negative correlations to colostrum or to fecal egg count? Is there any bad stuff that happens?
Yeah, so there is some literature out there that states when we start selecting heavily for post-weaning fecal egg count EBV, we can affect things like maternal weaning weight or even post-weaning weight. So I did a lot of that. If you were to go and look up, I think it's antibody response to CD&T in Katahdin lambs, you'll see there's a whole table there that describes some of those kind of production traits that you're talking about. And within the particular flock at Glade, we did not see those. So when I did the statistics to look at the averages of those groups, so whether you were parasite resistant or parasite susceptible, what we found that there was no difference in terms of your post-weaning weight EBV or your maternal weaning weight EBV or even your Katahdin hair index. So goes to show that at this particular unit, they're heavily involved in setting up the breeding scheme, so therefore trying to make it very robust and well-rounded while keeping the divergent post-weaning fecal egg count EBV in mind. So it actually is possible to select for post-weaning fecal egg count EBV and not see an effect of growth, especially when we're talking about, technically lambs that were sired by parasite susceptible rams should have better growth metrics, because a lot of times that's what we see with the Katahdin breed, is that rams that hang around for one reason or another that are really poor in terms of their post-weaning fecal egg count EBV are often really good in terms of other growth traits. So it's possible. Now, I say that with a grain of salt, because we haven't really done large scale analysis of that. That's only on a really small scale. But yeah, some correlations need to be put in place just to start to see if we do tip too far one way in terms of our selection for immune response. What does that cost us from a production sense?
Can you see Hamish Thompson's question, Kelsey?
I think so. Hold on. Okay, so I'm going to answer, I'm going to answer kind of the first part of that question. So commercially available for breeders. So you're talking about colostrum, like how can we commercially see high quality colostrum go on the market? I think that's what you're asking. And
I think he's probably asking that, like, commercially available tests, so you can test whether your sheep have got.
Oh, I see. Yeah. So we started that, actually, so I was going down the wrong path with that. No, so there's not. We tried Brix. Brix would be a refractometry. So it's just this little handheld machine that you could just milk out a ewe and then just take like a single drop of her colostrum and put it on a refractometer. And the refractometer measures mainly sugars. So there's something happening with Katahdin colostrum that doesn't give us a really good shoot side measure of, because like there are literature out there that states like, oh, if you have a Brix value of, I believe it's a 28, if you're a 28 or above, you have 50 nanograms per ml of IgG within your colostrum. And so I tried to do some of that within the Katahdin breed, and they were so atypical. They did not fall within those standards at all. So Brix was essentially out the window for producers, and I couldn't really give them a good, because that was the whole point. The ELISAs are time consuming and not everyone. So we wanted to be able to give you something where you could, in fact, start to sort high quality versus low quality colostrum. And so for now, the best thing that I can tell producers is if you see more dense yellow colostrum, I would be much more inclined to save and store that colostrum than I would kind of the milky white colostrum that we were seeing from the high grouping. Now, while IgG didn't correlate with that yellowness, I still believe that there's something going on that makes that of higher quality, whether it be from a nutritional standpoint or an immunity standpoint. I still think at the end of the day, what I can tell you from my research is if you see really dense, thick yellow colostrum, store that. And if you're milking out ewes and she's got really watery white colostrum, maybe if you have the opportunity, don't store that colostrum.
Did you do any correlation with genetic fat with anything? Like, did you have a fat EBV that you looked at?
No, I didn't do any sort of those correlations. But yeah, so that's another thing, it could be beta carotene, which is kind of the antagonist of vitamin A that I was talking about that could be driving the yellowness, or it could be fat. So we need to just do some basic, just nutritional looking at what is the composition of Katahdin colostrum. Because yeah, it might just be a compositional difference. Maybe low Katahdin ewes have more fat within their colostrum? The answer is I don't know until we start to analyze that from not only a nutritional sense, but also a metabolite sense.
Whilst we're on that, can I ask, did you look between ewe lambs and mixed stage ewes? Like, did you track it over years? Like the amount of IgG in the colostrum between a ewe lamb and a
Yes, because as you saw, so that first year we put parameters on ewe age, because as a ewe gets older, well, there's a fine line to all of this. So if you have a 9-year-old ewe, is she going to be producing her peak? It's not exponential, so the graph doesn't go like this. But from you being a yearling ewe to you being now a seasoned ewe and having a lambing, you are more likely to have more IgG within your colostrum. So that's kind of why we set some parameters there, because that's a well-known trait. So yeah, you could actually track her like IgG throughout her productive phase, and we could start to say, when does that threshold fall off? When do we start to get ewes that have kind of met their max IgG within colostrum, and now they're starting that decline of she's getting to a certain maturity where she's no longer putting the same amount of antibody within colostrum.
There's a long question there about Barbados black belly sheep. I'll let you go with that one.
So I am personally not familiar with Dr. Reyes. The Florida, I know some, like I know the breeds, but yeah, I'm not quite intent on talking about that too much. I'm assuming that if you're using those particular breeds, parasite resistance is going to be at the forefront. So I would assume that through projects like that, they would be taking some form of a fecal egg count genetic effect into play, but I'm not personally familiar with the project, so I really am not informed to say whether they are doing that or not. But if they're not, maybe you should encourage them to do so.
Another long shot, possibly, but I think a colleague of mine, and I haven't got it straight, so it's a risky question, but anyway, there seems to be some correlation between immune competence and heat resistance. Have you come across anything on that?
Yeah, so we see that with much breeds like that have developed, so breeds that have developed in traditional tropical climates are more likely to see parasitic antigen year round because larvae are going to thrive in that particular type of environment. So yeah, I would pretty confidently say that normally sheep that are more inclined to be parasite resistant would also be more inclined to be heat resistant. Now that's not a direct correlation. There's certainly it's not a 1-to-1 type of ratio. However, you could say that yes, in general, sheep that will have been kind of acclimated to those climates are more likely to be parasite resistant and vice versa.
Yeah, so it's an exposure effect there, right? Cool. Yeah. Thank you. Henry's got a question.
Hey, Kelsey. Well, folks, started off with some long shot questions. I thought I'd continue. We got a long shot double barrel question. I was wondering, like with your P work, have you, like, do you have any other, I suppose, like diseases over there, like say pneumonia or foot rot, that you found any correlations with WEC? And then the second part is, like, have you found in periods of, like say when the ewe is lactating and they lose that immune resistance, like the ones with lower WEC, is that when they're really turning on, or you find it's just a linear decrease for both the poor group and the high group?
So I'll start with the first question. No, I get that question all the time. People want me to do this with the pneumonia vaccine, and I do think that there should start to be some of those conversations. The problem is the pneumonia vaccine is a little bit less. It doesn't respond always in the manner in which we would like it to. So the efficacy of that vaccine is somewhat into play there. And that's not me saying that the pneumonia vaccine is no good. There's plenty of producers that use it and have great success with it. There's also some that sheep don't necessarily respond to that vaccination in the manner that we would like them to, and therefore they still, or they see a different strain of pneumonia. It's very kind of convoluted. So yeah, I believe that we need to be doing things like looking at pneumonia. My colleague, Dr. Weaver, switching gears from the pneumonia vaccine, he actually looked at incidence of mastitis and foot rot within those kind of ewe populations and wasn't able really to find a huge difference there between the groups. When we kind of talked about that, I think the idea was that Lee and his group at that particular production facility do such a good job of intensive management. A lot of those ewes that are the problem end up not staying around. So we might have incidentally maybe selected for ewes, like when he was milking out all of the colostrum samples, he actually was really happy with me because he was like, at the end of the day, I got to see all of my half bag ewes and they all loaded on the truck. So he was really excited by that, but maybe inadvertently in that production system, we're treating them with things like Zactran, which are going to be helpful against foot issues. And then we're also culling against half bag. So maybe that's why we're not necessarily seeing differences in terms of those diseases, because you're right. You would expect that that you, in theory, should be less likely to have a high incidence of mastitis, but that's actually not the case. We can't definitively say that yet, and we really don't know why. And then, what was your second question? I rambled on.
Just like when the ewe then like goes through lactation and it loses that immunity, like do you find the ones with low WEC, do they, like, is the response linear with both, like the rate that they lose their immunity during lactation, like are the low WEC and the high WEC ones both losing immunity, or do the low WEC ones lose less immunity?
Yeah, that's a good question. I would say if we're talking about age, because both of those ewes are going to age, I would say, which I haven't done this research, so I can't, this is just me strictly going on a limb. If you are more likely to have a high abundance of antibody within your biological fluids, you would more likely not lose, there would not be as steep of a decline, if that makes sense. So if we think about, we saw how kind of antibody in those U's is segregated like this at the very beginning when we're talking about colostrum. Well, there's not much of a floor to go from here. Those U's are just relatively low in terms of their IgG. So actually what more than likely would occur is those low U's, or the parasite resistant U's, we would see them actually drop off more so because there's more room to drop. Does that make sense? So if you're already low in terms of your IgG, it doesn't necessarily matter how old you get, you're more than likely still going to be low in terms of that value. Whereas the low Katahdin U's, or the parasite resistant ones, have more of that wiggle room to drop. So that 2.5-fold increase that I was talking about likely gets smaller and smaller and smaller as we see those U's progress in terms of age. Now, that is just me speculating. I have no data to validate that, but I would just think from a research standpoint that would probably be what occurs.
Just on Henry's question, so that periparturient rise, like we see in worm egg count, so immunity drops off as the ewe lambs. I think his question was, does a high WEC sheep drop faster than a low WEC sheep, or is that shift in immunity?
Oh, I see what you're talking about. You're not talking about colostrum antibody level. You're talking about periparturient egg rise. So yes, you're that would be the inverse then. So I was speaking from a colostrum standpoint. So when we're talking about antibody as those U's age, more than likely low U's would continue to be somewhat higher, but then as they age, they would kind of diverge to this point. From a standpoint of periparturient egg rise, so birth and lamb is actually considered a disease state because you have self versus non-self kind of competing with one another. So you're right. You, right around time of lambing, will go and decrease her immunity. Well, what happens then is parasites who also want to thrive, reproduce, and survive exploit this opportunity. And so they'll go and readily make way in mom. But mom has already experienced that antigen. So she's far less likely to show clinical signs of that parasitism. I'm not saying she won't, but those ewes who are in that particular production system are less likely just because they've experienced that infection before. More than likely, what we would see is lamb, who is right beside mom while she is actively shedding these eggs, is going to come behind her and pick up that infection. And that lamb that has never experienced infection before at all is going to really, really be impacted by those increased larvae present on pasture. So yes, it would be the opposite of what I was saying. Low dams are going to be more likely to shed more eggs, which are then more likely going to result in infective larvae. And lamb, who is already genetically predisposed to be more susceptible to that infection, is now far more likely to experience clinical signs of parasitism, whereas maybe the same lamb who was with a parasite resistant ewe is far less likely to experience clinical signs of that parasitism.
Mark Ragg asked, did you look at the difference between sire versus dam influence?
So we tried, how we kind of sorted it. So you can see the death loss is sorted on sire because we actually started with a sire effect. So everything in the very beginning when we're talking about response to vaccination was originally a sire effect. We were only looking at whether your sire was supposed to be a low PFEC sire or a high PFEC sire. Now, once we got physical data back on those lambs, we could then segregate lambs out and truly evaluate lambs' immune response to that vaccination. But from a colostrum standpoint, have we started to look at any effect of what sire has to do on that? No, we only looked at the effect of sire from the death loss perspective, as well as the response to vaccination. And so that was all the way back in my master's. So if you go and look up like my master's project, you'll see that the graph actually looks slightly different because they're grouped by sire. So we don't have individual lambs. We're now looking at the effect of that sire, versus now that we have enough data submitted to NSIP, we can post hoc go back and use a lamb's individual value. And the lamb's individual value is much more telling than what the sire influence of that mating is.
Cool. We're pretty close to the hour. Kelsey Keeling might be typing, I think, so we'll wait and see what Kelsey asks. But while we wait for that question to roll in, just want to really take this opportunity to thank you again, Kelsey, for jumping online with us, and it's been an international audience today, which is awesome. And yeah, it's great to be working across lots of different time zones, and thanks to Sophie for setting it up. That question has just come in from Kelsey. So once a lamb gets worms, is the only treatment to worm, or can you continue management practices, rotational dosing?
Yes, I highly recommend management practices. So especially over here in the States, I won't speak to you guys in Australia and New Zealand. However, what I will say is over in the States, I believe we're incredibly too reliant upon our dewormers and our anthelmintics. So we just expect them to do the job, but they were never meant to be a long-term solution, if that makes sense. So the efficacy of our anthelmintics wasn't a matter of if they were ever going to stop working. It was a matter of when are they going to stop working. Worms are, in fact, living things. And just like us, they want to thrive, survive, and reproduce. So therefore, they are actively altering their genetic makeup to be resistant to dewormers of specific dewormer classes that we have overused. So the answer to your question is absolutely yes. We need to be implementing things like targeted selective treatment, either through the 5-point check or from matcha scoring. We should not be mass deworming ewes all at once. We should, in an ideal world, only be treating ewes that require that treatment, or we should be rotationally moving ewes along with lambs to different paddocks or pastures to therefore decrease their likelihood of going back and ingesting larvae that they have spread out over pasture. So yes, all of those management practices that you already know about, I think in an ideal world, we would be continuing to use those. So that way we have judicious use of our anthelmintics and we can see them be successful well into the future.